morning, everyone. It's truly a pleasure to be here today, and the first time I think I've spoken something with physics in the title. So I'm not a physics person. I'm in fact a biologist. I hope that's okay with all of you. I work in areas of biology that sit at the interface of conventional fields. So we study genetics, development, evolution. We use computational methods to do that. So my goal today is to tell you a little bit about one specific project that has sort of been a long-term interest in my own research, but also to hopefully improve your understanding of genetics, development, and evolution more generally. So I'll introduce sort of those ideas of those fields early on, and then near the end, I'll be getting into a little bit like sort of one example from my own research where you can see how those pieces fit together. So as an evolutionary biologist, I am fascinated and intrigued by how an incredible array of biodiversity has evolved from a common ancestor. Charles Darwin was also equally fascinated by this, and I love the closing words of his Origin of Species, where he says, "From so simple a beginning, endless forms most wonderful and most beautiful have been and are being evolved." I think I said that flipped. And I share this fascination. And so as an evolutionary biologist, I want to understand how that process works. How have these changes arisen? How are they passed from one generation to the next? And that's the focus of the talk today. So there's different approaches that people take to understanding how evolution has occurred. Some people focus studies at the organismal level. So we have two different species here. Let's see, which screen, there we are, with differences in beak shape, and so this is a change that has evolved between these species. So there's a lot that we can learn just by looking at the organisms themselves. The form that an organism takes, and this is true of all living things, is encoded by its DNA. So this is a double helix representing DNA. DNA sequence is made up of 4 bases, A, T, G, and C, and changes in the composition of these sequences, if I can get this here. Maybe I'll just talk. The sequence changes in the composition of the sequences over time is actually what is causing the phenotypes or the traits to change. Okay? So there's also a field, and one that we participated in my lab, of evolutionary genetics, where we're focused on how DNA sequences are changing over time and what that tells us about evolution. So for a long time, there was sort of a black box between understanding how DNA sequences produce the phenotypes or the traits that they do. This is through the process known as development, and that's something else that we've learned as a field a lot about in the last 30 years, even more so than that. And this has allowed us to compare how development is different and similar between different species. Okay, so that's something else I'll be talking about today. And then the final piece, of course, is the concept that's more often taken as synonymous with evolution, and that is sort of natural selection, right? So which individuals within a population survive or are more successful at reproducing and contributing their genetic material, their DNA, to the next generation. Okay, so that's interactions with their local environment, interactions with their own genome, so sort of selection, drift is a random process, and migration all factor into this. Okay, so when I think about evolutionary biology, it's at all these levels and how they work together. Genetics is the term that we use to describe how inheritance works. This is where you start there because I suspect it's most familiar to you. As I can tell by your laughter, you've already appreciated that this figure captures the idea that you have genetic contributions that control phenotype in the father and the mother that are both contributed to the child and contribute and determine what it looks like. So some combination of the father and the mother, or the checkerboard shirt in this case. This is, of course, a gross oversimplification, but I think it gets the concept across quite well. Okay. When we think about this connection between genetics and the inheritance that happens within an individual family, and evolution and speciation and sort of those longer term processes, we're really just changing the time scale. So the next couple slides are designed to illustrate that. I think I will need to find the pointer on the slide for this. Okay, so these dots up here are representing individuals. So the red dot is this beetle named Mimi, and the lines, the green lines, are connecting Mimi to her parents that are in the generation before her. So up at the top is sort of present time, and as we move Lower in these diagrams, we're moving backwards in time. Okay, so Mimi has 2 parents. She also has 2 siblings. Okay? If we expand out and think about the whole beetle population at one point in time, each individual beetle has 2 parents that preceded it, sometimes siblings, sometimes not. They also have grandparents in the generation before that, and so on. And so we can trace the ancestry or the lineage from the present individuals back in time and form these types of networks. If we keep going on a larger time scale, thinking about the whole population, this process, again, is just continuing, and we can see this sort of messier sort of intermingling of lineages. If we back up even more, so this piece I showed you on the previous slide matches up to that blue square at the top, which is sort of a very small region of time. So that's very close to the present. So going further and further back in time, we just have, again, the same process. Parents are having kids. Well, the parents do pass on their genetic material to the children. There's also mutations that happen along the way that introduce changes and variation. So the kids don't look just like the adults, right? There's some combination, but there's also new traits that are arising over this evolutionary time. So if we go back far enough in time, in this case about 1 million years for these beetles, we come to places, so this process that I've just described is happening in this species of beetles, but also in this species of beetles. And again, when we go back far enough in time, there's a common ancestor, a common great-great-great-great-great-great-great-grandparent of both of these species that was in one population. Okay? So this sort of splitting of what in the past was a single species into 2 species is known as speciation. Okay, so one species is not turning into another species, but rather there's a branching process where a population, some individuals are acquiring changes that make them separate and different from the other individuals, and those ultimately become different species. Okay, so again, we go back even more in time, and we can see this process playing out over and over again. And of course, what's true for beetles is also true for all other life forms. So back in time here would be the common ancestor of all of the species shown in the present, which are the tips of the tree up here. Of course, not all species that have ever lived are still alive, and so we have extinction events represented in these phylogenetic trees such as this. Okay. So that's the evolutionary framework, and one consequence of this process is that the history of present-day individuals is encoded within its DNA. Right? So they got it from their parents, who got it from their parents, and so on and so on. So I like to say that DNA is actually a historical molecule that has a record of the history of not just that individual, but other members of its species, closely related species, the relationships among species, and so on. Okay. So I want to contrast that view of thinking about populations and splitting with the more common representation of evolution that you see. I assume most people have seen something like this before, right? I show this in my class, and then I quickly put this up. This is not how evolution works. I think this has done a real disservice to the general public for conveying how scientists think about change over time, which is evolution, okay? So it's not one individual progressing from one species or one form to another. There are populations that are diverging over time in this splitting process. So here's another way to represent that and relate it back to the shorter time scale that we're more comfortable thinking about. So at the top here, it says, this is not your family tree, right? Your great-grandfather didn't just give rise to your grandfather and father and you. Rather, you have something more like this, where you and your siblings share a common ancestor in your parents. You, your siblings, and your cousins all share a common ancestor if we trace these lines back from the present into the past here with your grandparents. You, your siblings, your cousins, and your second cousins share a common ancestor with your great-grandparents, and so on. Again, on a longer time scale, this is the relationships among different species. So we have here 2 different mammals, humans and another mammal from the cartoon. I'm not sure what that's supposed to be, but they had a common ancestor. If we go back even further in time, there was a common ancestor that gave rise to both amphibians and mammals. Even further back in time, a common ancestor that, as all of these different species and lineages move forward in time at the same time, right, in parallel, gave rise to fishes, amphibians, mammals, including humans. Okay. So that's the sort of evolutionary framework. We're going to switch and focus now back on what's happening in just that one generation, the really short time, and talk about the process of development. So when I said that we have, the mother and the father are contributing genetic information to the child to help it, to determine sort of what that child is, this is how that process works, right? So we carry our DNA, which is the genetic material, in the nucleus, which is this sort of purple area here, for females, the egg cells, and in males, the sperm cells. And hopefully this is not news to you that when an egg and a sperm come together, we can make a baby. And this begins with a single fertilized egg. Okay, so we have one cell in which the DNA, the genetic material from the mother and the father, has been combined. That cell will replicate its DNA and divide into 2 cells. Those 2 cells will each replicate their DNA and divide into 4 cells total, right? Each cell dividing into 2. And this process is just continued and repeated until the baby grows, and so does the mother in most cases. So we have a situation where multicellular organisms, animals like us that are made of multiple cells, generally have the same DNA in all cells, okay? So again, this process of growth is here. This is another way of representing that, the fact that all of our body is made up of these little units known as cells. Each cell has DNA in it, and because that cell was produced by making a copy from the original cell, it has the same DNA, minus any mutations that might arise along the way. Maybe I'll give you one little tangent and say sometimes those mutations that arise along the way during growth and development give rise to things like cancer, right? So cancer is actually sort of an evolutionary process too. Okay, so we have a body that's filled with cells that all have the same DNA. However, all of our cells don't look the same. They don't do the same thing. So here's an example of a few different cell types that would be found in a human body. We have different types of blood cells. Our skin cells have very different structure and function from these blood cells. Bone cells, we have cardiac muscle up there, epithelial cells, and so on. And so different cell types, at some point during that development process, right, we started with one cell, so that cell could only have one function and one form. But as the cell divisions were happening, decisions is a little too anthropomorphic, changes are taking place in those cells that give them different properties and different specific functions in the body. Okay, so how does this happen? This is actually one of the questions that first got me excited about biology and genetics, was thinking about this puzzle. It's really remarkable, right? They all have the same DNA, but they're doing very different things. The answer for how that happens comes down to a process known as gene regulation. So each gene, and in the human genome we have about 20,000 genes, to put that in perspective, has in essence a control switch. Sometimes I like the analogy of a dimmer switch. So each gene can be turned on or off, and it can be turned on to a different level, right? A different degree of brightness, a different amount of that gene can be produced. And its differences in the sort of positioning, which switches are on or off. So these little cartoons here, each one would represent a gene. The red and blue would represent the part of that gene that encodes for the protein, which is the molecule that actually does stuff in the cell. And the beginning, the symbols at the beginning, the arrows would be genes that are turned on, and the X would be genes that are turned off. So in cells that are kidney cells, there are a different set of genes turned on, producing their gene products, than there are in skin cells. Okay? So the regulation of which genes are on when and where and how much of that gene is being produced is really a key step for development. And by the end of the talk, you'll see it's also a key step or a key process for evolutionary change. Okay. So if you're not used to thinking about molecular biology, I like to try and use this analogy as well, which is that some people will say that the genome is a blueprint for living things, and that's true. I think there's a little bit more nuance that we can even pull out of that. The proteins, which are, again, the molecules that are made from our genes, function like tools, right? So we can have a set of tools that can be used to build many different things. You can use a similar set of genes, and if you use them in different ways, use a hammer in this position for one species, if you will, and a saw in that position for another species, you will end up with different forms. Okay. So again, the analogy here is that proteins are the tools. Other parts of the genome that contain sequences known as non-coding sequences that control regulation of when and where and how to use those tools, that would be sort of the blueprint, and they work together to produce the phenotype, the trait, the house in this case. Okay. So there's a few experiments I'm going to tell you about in the next couple slides here that were really instrumental in helping the research community reach this understanding of how development works and how changes in development and gene regulation can contribute to evolutionary change. So the first, or one of the first, was an observation that when we had mutations that changed where a gene was expressed, we saw changes in the phenotype. So in this particular example, this is a close-up picture of a fruit fly's head. That's what you're looking at there. So these are its two eyes. I keep losing this pointer. An eye here, an eye there. These are the antennae. This is the proboscis, or sort of the mouth part. When a particular gene's expression was changed, was in fact turned off in the antennae, what was discovered is that these antennae now, instead of developing as antennae, developed into legs. So this is a mutant fly here that has legs where the antennae should be. Okay? This is a class of gene known as a homeotic. It's a homeotic mutant. It's a transformation of one part of the body plan into something else. Here's another homeotic mutant. This is what happens when you turn off a gene that is usually on in the sort of middle part of the fly here. It's thorax that includes the a part of the thorax, which includes this haltere, this balancing organ. And what happens when you change that is that the fly, the fruit fly, which normally has only 2 wings, develops 4 wings. Okay? And it actually develops a whole second thorax region. Okay? So it's a transformation of one part of the body plan into a different part of the body plan. And interestingly, evolutionary analyses have shown that this haltere, this balancing organ, is in fact a modified version of the hind wing from the ancestors. Okay, right? So dragonflies are 4-winged. As they split, as sort of the evolutionary process unfolded, modifications evolved to that hind wing that gave rise to the haltere. So genetically, a couple switches can actually restore sort of the old developmental program. Okay. All right. Another kind of unexpected finding that really had a big impact on how we think about development came around 1995. And this was a time when we were first learning to be able to turn genes on and off in a controlled manner, to move genes between organisms. And the finding was, I guess before I give you the punchline, I'll say up until this point, it was thought that elephants looked like elephants because they had elephant genes, and snakes looked like snakes because they had snake genes, and cows looked like cows because they had cow genes, and humans looked like humans because they had human genes. So what was really surprising was the discovery that those sets of genes in these different animals, although some of it goes much deeper than that, are remarkably similar. Okay? So these 3 pictures at the top show eyes in humans, mice, and fruit flies. And there's a gene that exists in the genome of human, mice, and fruit fly that has very similar DNA sequence, right? So genes are made of DNA sequence. It makes a very similar gene product, the protein. And when you mutate or disrupt that gene in each of these 3 species, eye development is affected. Okay, so this gene's name is PAX6. PAX6 mutations in this gene are associated with improper eye development in humans, the loss of eyes, in fact, altogether in mice during development, and then also in flies. Okay? So this was one of the first clues that, again, the function of a gene might be conserved over evolutionary time. So not just the DNA sequences being passed from from parent to children over and over again, but the function of the protein can be the same despite many changes in the organisms that carry it. Another interesting sort of test of this, or piece of this, was an experiment that was done with somebody actually that I was a colleague with at Wisconsin. He did it as a grad student, and I knew him after he graduated as a postdoc. And that was to take the gene that controls eye development in a mouse and put it into a fly. And so they did that first, and what they found was if they put it into a fly that had a mutation in this gene, so the fly's gene is not functional, and we're going to add to it the mouse version of that gene, the fly's eye was restored. so back to normal. If you turn this gene on in cells where it usually isn't, such as the antennae, this gene was also sufficient to turn the antennal cells into, this is not a functioning eye, but eye-like cells, okay? So the things that were profound out of this were, number one, that there's a lot more conservation of the set of genes, what we now know or describe as the toolkit, in animals especially, and that changes in how they're used, when, which cell, what point in development, how much of that gene product is made, can have major, major changes in morphology, right? And may be responsible for a lot of the differences we see among species. So that was the hypothesis. We now have mountains of data that support that, lots and lots of data. I'll show you one example of the type of data that does support that here. It's a little bit hard to see, I can tell. So we have at the top is a butterfly species. And I want to focus your attention on this sort of concentric rings of color here, which we know is an eye spot. So this is just a picture of the eye spot on the butterfly. What you see in the other panels, here, here, and here, are what we call stainings, showing where the products of certain genes are. So our body makes antibodies to fight off pathogens, bacteria, viruses, and so on. Researchers have harnessed the ability of an antibody to recognize a specific thing and use that to recognize a specific protein of interest. So it's now become a research tool, antibodies. And we can connect that antibody to a fluorescent label, so a colored dye, in essence, that we can see. So when we do that, we can look at which cells, so again, this butterfly is made up of cells, which cells are producing a particular gene product that might be making some cells have yellow pigment, and some have black, and some have white. So what I hope you can see, even if faintly, is there's sort of this ring of green here that in fact corresponds to the location of the ring of yellow. Here there's a ring of purple that corresponds to the black, and this is just those two rings combined, so you can see how they fit together. So this was a suggestion that these particular genes that we're marking with our tools here, our antibody tools, may be specifying the development of black versus yellow cells. So to test this hypothesis, the researchers took advantage of a mutant where the shape of the eye color was sort of jumbled and disrupted, and then looked at the expression of these same genes in that mutant and found that the expression of those genes was similarly jumbled and messed up. Right? So this was pretty compelling, circumstantial at least, evidence, correlative evidence, that changes in expression of these genes were associated with changes in the development, and therefore ultimately the phenotype. It also suggests then that maybe this is one important mechanism by how evolutionary changes have occurred. And this has been tested. Again, many, many, many studies support this. I'm just showing you a selected set today. Here we have eye spots from different species of butterflies, and they come in a remarkable diversity. You may have seen there's like an alphabet eye spot calendar. If anybody hasn't seen it before, it's kind of cool. We can mimic that by looking at gene expression patterns. So I'm not sure I said it explicitly, but these gene expression patterns that you're seeing, these colored rings here in many cases, those are detected when this butterfly is still in its cocoon, so the pupal stage. So before the colors are actually forming, we can see where they will form based on those expression patterns. So when we look, in fact, now at different species of butterflies that have their own patterns of eye spot pigmentation, we can see corresponding changes in expression of these genes that, again, match up with that, suggesting that evolutionary changes in DNA sequence have altered where these genes are turned on and expressed, and in turn that has altered development, which has altered the phenotype that we see. Okay. So pigmentation, I think, is a great trait to study if you want to understand how evolution works. So again, if you're interested in the specific mechanisms, it's really hard to study things that are very far apart. Right, so if I wanted to know how the fish's tail was different from the butterfly's wing, there's so much that's changed. It's really hard to address that. Or how a plant leaf differs from a giraffe neck, right? I mean, these have evolved from a common ancestor, but tackling that is it's a bit much, right? Because this has been a step-by-step process. So a lot of the information that we have that leads us to our models that we trust, that we believe are best supported by the data, comes from studying more closely related species. So as you can see in this picture, pigmentation is highly variable in many different species. In fact, I would argue that it may be the most variable trait within and among species. We can think about even our own species, right? Skin color is incredibly variable throughout the human population. Okay, so in fact, pigmentation it's also something that affects the way organisms interact with their world. So it's likely to be subject to natural selection. And it's pretty too, right? Often, so it's sort of visually compelling. And I kind of say that jokingly, but especially when talking to people who don't live and breathe and think all the time about biology and molecular biology, that's important, right? To have something that's relatable. So I think these case studies of pigmentation are really important for a broader conversation about how evolution and development work. Okay. So as I said, pigmentation does affect the way organisms interact with their world. For different species, different ecological factors seem to be controlling most how changes in pigmentation arise. So in some cases, camouflage or crypsis seems to be really important. That's illustrated here by this picture of rock pocket mice. So this is a system that Hopi Hoekstra, who's now at Harvard, has studied in great detail, figuring out the genetic mechanisms responsible for these differences. It's a evolutionary interesting example because the difference between these lightly pigmented mice, which blend in with the brown sandy environment, and the darkly pigmented or colored mice, which blend in with these dark habitats, have evolved in only about 10,000 years since these lava flows have occurred, which created the black substrate. So it's a more recent change that can be investigated. We can actually, in the lab, cross together the light mice and the dark mice. This allows us to look at how those colors are passed on to their children. That tells us something about what's controlling them genetically, and it makes it a very tractable system. Okay, so camouflage can be important. Warning coloration is another thing that has been proposed to be driving evolution of pigmentation. Thermoregulation, especially in insects, right, where the amount of UV that's affecting you is going to change your body temperature. So how dark you are, if you're in a climate that has less UV or is colder, right, you might want more dark coloration that will absorb more of the light and the heat. Sexual selection is another key role that pigmentation has for organisms. Right, so thinking about the very colorful male peacock tails, right? They are thought to have evolved as they are because of female mate choice and preference. If females will preferentially mate with those that have more elaborate colors, those males with more elaborate colors will be more successful at passing on their genetic material to the next population, leading to more children with elaborately colored tails, and so on and so on. Okay? All right. This is also true for fruit flies. I think that I am in love with their pigmentation patterns, but maybe they're not as broadly exciting to you. But there is a lot of variation when you look at fruit flies close up. Some have, this is the abdomen of the fly here, right? So sort of the lower part. This is that thorax I pointed out before. This is their head. And so you can, and these are, I hope, of course, wings. You can see they have different spots, different stripes. Some are darker, some are lighter. There are a number of selective pressures that have been invoked for why changes in pigmentation might have evolved. These again include camouflage, thermoregulation, desiccation resistance, or resistance to drying out in less humid environments. Resistance to parasite infection is also interesting. So these pigments that appear as color also change the structure of the cuticle. That's actually why they affect the desiccation resistance. They change the water permeability of the cuticle. They also can change the ability for some parasites to get in. They can protect against UV radiation, which is something that can cause mutations. And then again, sexual selection related to courtship behavior and mate choice. This final piece is something I won't talk about today, but a grad student in my lab is studying. There's a species, actually a number of species of flies that have evolved a male-specific spot on their wings. And the same species that have evolved that spot on the wings, as part of their courtship display, show that spot off, whereas very close relatives that don't have that spot don't show off their wings during the courtship. So there's some, again, circumstantial, and we're trying to get the sort of functional genetic evidence linking those two traits. Okay, so this is where we're going to get to a little bit of some of the original data from some of my own work, and I've sort of reached back a little bit here for this story, and much of this was done actually when I was a graduate student at the University of Wisconsin with a guy named Sean Carroll. I don't know if some of you may or may not know. So Sean Carroll is currently the Vice President for Education and Outreach at the Howard Hughes Medical Institute. He's also written a number of books about evolution and development and science in general for the general public. So if there was still a Borders down the street, we could go there and find books that he's written there. So if you're interested in learning more about this field in general, Sean B. Carroll would be a great name to look up. They've won a number of awards. They're very well written. Okay, so this is what we refer to as a wild type fly. Wild type in genetics just means that this is the form that you would find most often out in the wild. Right? We're not very creative in that name. So here is the fly, which may look more familiar, right? So head, thorax, abdomen, wings, legs are sticking out from the side. This is 3 of the segments on the back or the dorsal side of the abdomen. Okay, and that's what I'm going to be showing you for the next few images, is just that dissected piece of the abdomen. So you can see that this species, which is Drosophila melanogaster, the most commonly researched fruit fly, and perhaps one of the most common, well, one of the first, or the first genetic model system, has this dark pigment stripe at the back of each of these 3 segments, and then some lighter pigmentation in front of that. Okay, before I show you the next image, I'll just mention that as a geneticist, the way we learn about how genes work is to break them. Okay, I like to, again, use the analogy of a mechanic trying to figure out how a car works by opening the hood, reaching in, pulling out a part, trying to start the car and see what it does. Right, so you pull out some random wire, you try and start the car. Does it start? Well, yeah, it still does. Okay, guess it's not essential for that. You turn on your headlights, oh, they're still on, not involved. You try and turn on your windshield wipers and they don't move. Right, and so now you figured out that that random piece you pulled out affected the windshield wiper function. Okay, so that's effectively what geneticists are doing. We introduce mutations, or we take advantage of mutations that arise naturally that disrupt what an organism looks like, and then work backwards to figure out where that mutation is, which gene has it, and then infer what it does. Okay? In the early days of genetics, especially Drosophila genetics, so that we're talking about the 1900s, genes were given names based on their phenotype, based on their mutant phenotype. So what the organism looked like when you broke the gene is what name you gave the gene. For a little bit more perspective, again, this work was being done in flies around the early 1900s, like 1900 to 1910. We didn't figure out the structure of DNA till like 1950s, right? So we didn't know what hereditary material was, but we knew that there was something that was passing this information on. So that some of this terminology was actually coined before we knew the molecules that had those functions. All right, so one of these very, very first mutations that was ever isolated, this was in Thomas Hunt Morgan's lab. He's known as the father of Drosophila genetics. And I guess as a personal side note, my daughter is also named Morgan, and that might not be fully coincidental, was a gene, a mutant that he and his graduate students named yellow. So Thomas Hunt Morgan has won a Nobel Prize, Calvin Bridges, Sturtevant, basically at least I think 3 of his other graduate students have also won Nobel Prizes for their work on early Drosophila genetics. This is where we learned about genetic maps, that genes were on chromosomes, how recombination works, how sex determination works. A lot of the things that we take for granted were first figured out by studying the fruit fly. Okay? So next time you kill one in your kitchen, just say thanks first. Okay? All right. So we have these yellow mutants that were named yellow because they look yellow, right? Maybe not the most creative naming system, but one that works. Another early mutation was called ebony because the fly's pigmentation was darker, okay? This does lead to a complicated thing for me today because the normal function of the yellow protein is to make black pigment, right? You break the yellow gene, the black pigment goes away, the fly looks yellow. So yellow makes black pigment. The function of the ebony gene is to make yellow pigment. So yellow makes black, and ebony makes yellow. Got it? Okay. So that's important to know as we go through here. We also, or I did, as a graduate student, looked at flies that were mutated for both of these genes. And what I saw was that not only did it have kind of this intermediate color, it also lost its stripes. So this was informative to me because it said that in this yellow mutant here, the stripe pattern we see is being controlled by the ebony gene, right? Because if we take ebony away, the stripes go away. It also told me that in the ebony mutant, the stripes that we see are being controlled by the yellow gene. Right? We take the yellow gene away, and the stripes go away. So they're both contributing to making this stripe pattern. There's a third gene involved, tan, that I didn't know about in grad school, but we've been studying a lot since, that also fits into here. You'll see it pop up on a slide in a minute. Okay. Now, I told you before about our antibody tools that allow us to look at where gene products are, where these genes are expressed. What you're looking at here are two of these segments. Right, so like these two, for example, earlier in development. So again, they're still in their cocoons, although we call them pupae. And they're developing. The adult form hasn't fully developed, but we can already see this pattern of expression in the yellow gene that is going to determine where those black stripes go. Okay? Ebony, I also did that experiment, and it's expressed actually more broadly. So this is a closer picture that's only sort of one region like this, okay? So we can look at the expression patterns. I can look at the mutant phenotypes to infer what these genes are doing. We also know something about what the proteins they encode do biochemically. So the pigments in a fruit fly, there's a yellow, brown, and black pigment. They're all produced by biochemical reactions that function in the same pathway. So the fruit fly, as part of its diet, gets tyrosine through its food that it eats. Tyrosine is one of the amino acids. That the function of yellow, ebony, tan, as well as some other proteins, basically catalyze reactions to turn one molecule into something else that ultimately form black, brown, and yellow pigments. Okay? The reason I mention this pathway is that because it's all working off of the same precursor, if we change expression level of yellow or ebony, or tan in fact, we end up shifting the balance between these different pigments. So if I make more ebony, this pathway predicts, and in a minute I'll show you, you get more yellow pigment produced and a reduction in the black and brown pigments as these molecules are sort of used in a different way. If we upregulate yellow, although this one's more subtle, again, you'll see the picture in a minute, we've produced some extra black pigment at the expense of some of the lighter pigments. Okay. So we can use our knowledge of the biochemistry, the genetic data, and also this expression data to think about how development is making that simple stripe. Okay. So I mentioned before that if we can change expression, we can see what the genes are doing in a more, I guess, powerful way. Drosophila geneticists have been developing tools to study and manipulate genomes for the last 100 years. I've said that as well. One of these tools allows us to turn on virtually any gene we want in any cells that we want at any time that we want during development. Okay, so this is a really powerful system. That's the type of system that I've used in this experiment. What you see in this first panel is actually a picture of a Drosophila pupae, so still in the cocoon, although I've pulled it out. This would be its thorax here. This is the abdomen down here. The head is cut off, so you can kind of match these up like this. The green color that you're seeing actually comes from a protein that was found in the jellyfish genome. So jellyfish are naturally fluorescent. Researchers have figured out which gene causes that fluorescence, and the sequence for that gene has now become another tool in molecular biology that we can use to see where other genes are expressed, okay, just by putting it under the microscope and looking at it. So in this particular slide, this is here just to show you what part of the fly I'm going to be turning on the different genes in. So in the first experiment, I overexpressed the ebony gene. I turned on ebony where it normally isn't and at higher levels than it usually is in this stripe right along the center of the back of the fly. And what I hope you can see, it's a little grainy, is that the black pigment is gone and there's sort of yellow pigment in its place. Sort of like we've erased the black pigment. I did the same experiment with yellow, and what I observed was that there's a broadening of the sort of dark pigment in the middle. It's not quite as dramatic as ebony alone. And so I thought, I wonder if the wild type or the regular expression of ebony is interfering with the ability of yellow to change these patterns. So I then expressed yellow in a fly that doesn't make functional ebony protein, and now I do in fact get this dark stripe. Okay? So by manipulating expression of yellow and ebony, we can switch pigmentation around. We can create new patterns, we can change the relative abundance of the two types. So if I can do that in the lab, the suggestion is that what's happened in nature? Okay? When we look at different species that have evolved different types of pigmentation, such as those shown here, this species has stripes, this species is all black, this is sort of a lighter color. Again, this is a close-up of their abdomens. We can look at, I did look at, where the yellow protein was expressed in all of these species. Again, using that antibody tool. And what I found was expression was different in the species, and it was different in a way that correlated with the patterns of pigmentation that we saw in the adult flies. Okay. So this suggests that we can actually back up the problem, right? So if my question is, how did these different pigment patterns evolve, I now can ask, how did these different patterns of yellow expression evolve? Right. Okay. In other pairs of species, there are changes in ebony expression. Again, ebony promotes yellow pigment and inhibits black pigment, but not expression of yellow. So not all species are doing this the same way. Most often, when we see differences in pigmentation, so here are two different, actually this is just male and female wings of the same species. The males have this spot of dark pigment, which is a melanin, at the tip of their wing that the females don't. And when we look at expression of the yellow gene, again, the yellow color here is showing us where the yellow gene is, which cells it's expressed in. So each of these sort of structures would be 1 cell, 2 cells, 3 cells, 4 cells, just for perspective. Purple is showing you where ebony protein is in the spot that will develop into that pigment spot. Yellow expression is up and ebony expression is down. And again, that's analogous to the experiment I did in the lab where I manipulated those two changes and saw an increase in the black pigmentation. Okay. So this is, again, supportive of this idea that genetic changes have altered expression of these genes to give rise to the changes in pigmentation, in phenotype. Okay. So as a molecular biologist, I like to really understand how things work in the finest level of detail possible. I'm going to take you through a little bit of that, a little bit deeper here. And to do that, I need to tell you a little bit more about how these genes are regulated, how this process works. So again, we have DNA sequence. Instead of writing out the letters here, I've just used a line to represent it. Part of the DNA sequence is known as, so the exons, this is the part of the sequence that determines what the protein will look like. Again, the protein is the product of the gene that actually catalyzes biochemical reactions in the cells, forms the membranes of the cells. It does everything. It's sort of the tool. There are other regions of DNA nearby, known as cis-regulatory regions, which again are DNA sequences, and they bind to other proteins in the cell. Okay, so these proteins were now made by other genes, and when they interact with each other, they produce this gene. Okay, so this is sort of turning it on at the molecular level. So the two pieces I want you to think about are the cis-regulatory sequences, which are connected to the gene we're talking about, yellow in this case, and then the transacting factors, which are the proteins encoded by other genes that can come and that do come and regulate it. So this sets up a puzzle, because if we observe that a gene has changed expression between two species, we can't say anything about how that expression change occurred. It could have occurred because the cis-regulatory sequences of the gene changed, or because the transacting factors that regulate that sequence changed. Okay? So this is, I'm going to go through the schematic first, which is the logic behind the experiment that I will then show you the results from. So what we did was to take the yellow gene from this, from one species, a species that has a pigment pattern that's different from Drosophila melanogaster, and move it into Drosophila melanogaster. Okay, that's the basic design. So if we have a gene with, again, this is the coding sequence we're going to detect where it's expressed. This is the transcription factor protein that can regulate it. So if this transcription factor protein happens to be present in cells in this region, so these squares now are sort of a hypothetical tissue made up of cells, then wherever that blue protein is produced, the yellow gene should be turned on. Okay. If we take, actually I'm getting one step ahead of myself. if there are changes in the cis regulatory sequence of this gene, often they will cause it to bind to a different transacting factor. Okay? If that transacting factor has a different expression pattern, such as this direction, now we have changed the expression of the yellow gene. Okay? On the other hand, it's possible that the cis regulatory sequences stayed identical to the original copy, but what's changed is the expression of this regulator, so of this purple factor. So now blue. The blue has gone from a horizontal to a vertical stripe, and this also caused the yellow to go to a vertical stripe. Okay? So we can see that the stripe has moved from horizontal to vertical, and we won't know from that information alone which of these two mechanisms was responsible. Okay. We can sort this out by doing the experiment I was already starting to introduce, which is to move a gene, the yellow gene, from a species with one pigment pattern, so this sort of fully pigmented dark species, into Drosophila melanogaster, which normally has this striped pattern. Now, this is not a wild-type Drosophila melanogaster, but one that is mutated for the yellow gene that it carries. Okay, so it's sort of a blank slate, and we're giving it its only copy of yellow. If the changes in this gene that have evolved between these two species are located in the cis regulatory sequences of yellow, then when we move this gene into melanogaster, we're moving those changes with it. And that's going to produce a pattern of expression that is sort of throughout each segment the same way it is in the species we took the gene from. If, however, this cis regulatory sequence is the same between the two species, and what's making the different expression pattern is some other factor that we aren't moving from one to the other, when we move this gene over, we should get a pattern that looks like this species, Drosophila melanogaster, which is the stripe. Okay? So that's the logic behind the experiment. Here's the results from the experiment. So this is, as a scientist, we like controls. This is the staining or the signal of yellow that we get with our antibody when we look at a mutation, a mutant that shouldn't have the protein present. Okay. This is what we get when we stain the Drosophila melanogaster species. This is the one that has the stripe. And this is expression of the melanogaster gene put back into melanogaster. So this is a negative control, and this is a positive control. We know what we expect in both cases and confirm that we see that. We then take species. This is the one that was shown on the last slide that's very darkly pigmented throughout. When we look at yellow expression in that species, it is bright and sort of throughout the segment. When we move its gene into Drosophila melanogaster, we see the same pattern. So by moving this gene, we've moved the pattern into Drosophila melanogaster. The same thing with another species, the one that wasn't quite as dark but still had pigment throughout its body. Okay. We did a similar set of experiments with the wing spot species. So we've taken the yellow gene, moved it into melanogaster, which doesn't have the wing spot. If the changes are in the cis regulatory region, this experiment should give us the wing spot the wing spot expression in a species that doesn't usually have it. If the changes are somewhere else in the genome, then we should get a pattern of expression that looks like the species we moved it into, which is Drosophila melanogaster here, no spot. And that's the result. So again, with both the abdominal pigment patterns as well as these wing pigment patterns, what we're seeing is when we take the yellow gene, including its regulatory DNA nearby, and move it into a totally different species, number one, it's expressed and it works. Number two, it retains the pattern from the species it came from. So this tells us where in the genome, right, this genome of, combined with 20,000 genes, where these changes are. Okay. Oops. So we now know a lot about these sequences that we're moving, these non-coding sequences, the ones that are controlling the gene expression, when, where, and how much of genes are turned on. These little boxes here are the representations of that. So the line, again, is representing DNA sequence, and the boxes are just highlighting parts of that DNA sequence that activate expression in specific regions of the body. So the fruit fly's body. So there's a wing spot, a wing blade, different parts of the abdomen, which is the part they're referred to as body. And when we look between different species, which is what these three lines are here, different changes have occurred in these DNA sequences. So for instance, the spot has been lost because mutations have occurred in this set of DNA that make it make it unable to bind to those transacting factors and activate expression in the spot. In the other species down at the bottom, mutations have occurred in the part of that DNA sequence that was telling this gene to be active in these bottom two segments in males. Right? So that has changed that. So what we have is a system in which all these independent DNA sequence modules are controlling expression, sort of lots of switches. So it's not one dimmer switch for a gene. There'd be a dimmer switch that can be independently controlled in the wing spot, in the wing blade, another one in the body, another one in the legs, another one in the bristles for this gene. And this is a common feature of how genes work in multicellular organisms. So that was quite a realization. And it's now been seen again over and over again that changes in these switches, these non-coding regions that are telling genes when and where to be turned on and how much product to produce, are underlying this diversity. So I have just two other brief examples to point out to you that I won't go into in any detail, simply as other examples of changes in these regulatory sequences, cis-regulatory sequences affecting phenotypes. So one comes from sticklebacks, so stickleback fish. Marine populations of sticklebacks have heavy bony body armor on both sides of their body, but they have invaded freshwater areas multiple times independently around the world. And virtually every time they do, there's a reduction in the body armor. So the loss of body armor appears to be adaptive in freshwater environments. The genetic changes responsible for that loss in body armor have been well characterized at this point. And changes, basically they are cis-regulatory changes, so that the whole function of this gene is not lost, but rather function of one of its switches that turns it on a particular region of the fish. The same gene in this particular case that's affecting body armor in fish, this is more hypothesis. It hasn't been proven with sort of functional testing, but seems to be potentially involved in the evolution of the human foot and how it's evolved differences from the chimp's foot. Right, so that the enhancer has different functions in humans and chimps, and that may be contributing to the difference in morphology. Okay, there's again lots of examples like this that I could go into. The final one, just so that you don't leave here today thinking this is only happening in animals, because I am a little animal-centric, I admit, is corn, teosinte. That's also good to end at maize, right, when you're at Michigan. So this is maize here, the plant, not the color. This is maize. This is a close-up of maize or corn. And this is teosinte. So teosinte is the wild ancestor of maize. So humans have domesticated maize over the last 10,000 years by selectively breeding plants that have characteristics that they deem to be desirable. Okay? So through that process, this slide doesn't show it well, we've had an increase in the size of the kernels in the ear, the number of kernels. The wild relative has actually like hard seeds outside the kernels. You couldn't just pick it up and eat the kernels, right? There's a hard seed around it. So that has changed. And then what this slide was meant to highlight is sort of the branchiness or bushiness of the wild plant versus the domesticated form that we grow nice in rows, right? And so the gene that is responsible for this change in branching patterns has been identified. It's called teosinte branched 1. And changes in its regulatory sequences that alter which parts of the plant are expressing this gene are what's responsible for some of these differences. So that's the last slide, and I guess I would like to wrap up by hopefully conveying an excitement and amazement that I share with what we've been able to learn about genetics, how they control development, what role gene expression and regulation play in this process, and how by using living organisms that are around us today, we can actually start to do experiments in the lab that can disentangle the evolutionary changes that have occurred and help us understand how the diversity of life around us has come to be. So with that, thank you, and I'll be happy to answer questions, but I think we have a break first.